Okay.
Perfect. Hi, everyone. My name is Tolly. I'm a PhD candidate at the University of California, Davis in the graduate group in ecology. And today I'm excited to share with you some of the results of my first dissertation chapter, where I used DNA metabarcoding to study the diet of coyotes in San Francisco. So everyone here knows that animals live in cities, and they have found ways to either mitigate, adjust to, or exploit the novel resources and conditions that are present in the urban environment. And one of the most basic elements of a species ecology is what it eats and how it eats. And so it follows that one of the most widespread behavioral adjustments that we see in urban animals is this shift in their diet and their foraging behavior. And this happens because urbanization really dramatically alters the quality, the quantity, and the distribution of resources in the environment. And in particular, human areas provide a lot of food for wildlife, sometimes intentionally through backyard bird feeders or direct hand feeding of wildlife, but also inadvertently through garbage and compost. And some species that eat these human-provided foods see benefits like increases in fecundity or improvement in body condition, but there are also many disadvantages to these human food subsidies. For example, Jeremy Lyons et al. In 2017 found that urban chipmunks had reduced activity and lower locomotion compared to their non-urban counterparts. A study on raccoons found that those with more access to human-provided foods were heavier and had elevated markers in their blood for blood glucose. Maureen Murray, who is an organizer of this conference, found that coyotes that were consuming compost were exposed to toxins and were in poor condition. And then another study by Adelman et al. In 2015 found that finches that were spending more time on these backyard bird feeders were also more likely to become infected with bacterial pathogens. And in urban areas, reliance on these sorts of foods can bring animals into regular contact with people. And this can happen through changes in their movement, through increases in boldness or habituation. And all of that can create the possibility for direct attacks on people or their pets, property damage, or even erratic behavior. And so in this video, which my parents scoped out on their local next door newsfeed, you can see this play out with one of the urban animals that's sort of been in the spotlight for urban human-wildlife conflict. So I'm just going to go ahead and play it. And if you listen, you will hear a woman scream. There might not be sound in this, but she screams because that coyote actually runs into her bedroom fully after the dog. And what I find fascinating about this video, I'm going to try to play it one more time, is that if you look, there's two coyotes in this video, but they don't behave in the same way. While one runs in, the other one nopes out right about there and says, that's it, I'm done, I'm not going in. And I think that's fascinating because there's this difference in their boldness, there's this difference in behavior, and we know that individual variation exists, but we don't always understand how it happens. In this case, what leads one of these individuals to act in this way that can lead it into this conflict type situation, and this is not the same for the other individual. And so not only do we not understand really the mechanisms that lead to this inter-individual variation, we don't even really understand how widespread it is in cities. And that brings me to San Francisco, which was recolonized by coyotes in 2002 when they first appeared in the Presidio, which is that big park in the bottom left corner. And what I like about this photo is it really shows the heterogeneity of the city and how different life as a coyote might be depending on where you live in the city. Expectedly, coyotes live here in the Presidio, in Golden Gate Park, in Twin Peaks. These are like big, large parks that you might be familiar with, but they also live here at Coit Tower, at Corona Heights, and at Bernal Hill, where most of the land is totally developed. And for a short stint in 2021, they also lived here on this solar panel project in the middle of the city. And so lots of diet studies have examined urban-rural differences in diet, but what they tend to do is group urban together as a single category. But when we look at this landscape and see the heterogeneity of it, we know that individuals can differ in their behavior. We might anticipate, well, there probably actually are these individual differences in diet across the landscape. We also know that conflict is not equitably distributed across the city and that diet can underpin conflict. So it follows that we expect diet to maybe vary across this landscape as well. And this is what I'm really interested in, is how the heterogeneity of the built environment coupled with the widespread availability of human food resources sort of coalesce to influence coyote diet. Specifically, what are coyotes eating in San Francisco, and how do diets differ across the landscape and among individuals? Traditional approaches to studying diet are either based on observation or on morphological analysis of prey remains in scat, but this requires taxonomic expertise, is pretty time-consuming, and can't always distinguish between species in the scat. And all of that is because there's this reliance on the undigested remains of prey in the predator's scat. And importantly, this is kind of a problem in these urban landscapes where there's a lot of anthropogenic food, because something like, let's say, a chicken nugget is not really going to leave a lot behind in the scat that is easily distinguishable. But what it will leave behind is chicken DNA. And so to combat these drawbacks, I used a molecular approach to studying diet called DNA metabarcoding, or diet metabarcoding. And in metabarcoding, we use the DNA present in a scat to quantify the diet. And so we look for these molecular traces of diet, which therefore is not relying on these undigested parts in the scat. And so it can get us down to high resolution species level diet frequently, and it can really capture those anthropogenic components of the diet. So briefly, the way DNA metabarcoding works, we first extract DNA from a sample, like a scat, and we extract both the host's DNA as well as the DNA of everything that it ate that is in that scat. Next, we use a primer to target and amplify a specific region of the DNA. We take this amplified PCR product and we sequence it, and then we use those DNA sequences to barcode or identify the taxa from which they originated. And in this study, I used a short section of the mitochondrial 12S gene, which targets vertebrates. I did also use a different primer to target plants, but today I'm only going to be speaking about the vertebrate diet data. After sequencing, I removed all scats that had a low amplification success using a conservative filtering approach. Monica Serrano, my colleague, genotyped these samples to assign them to individual coyotes as part of her project on the population structure of the San Francisco coyote population. And then I quantified diet using both percent of occurrence, which is aptly abbreviated POO, which is just frequency of occurrence rescaled to sum to 100, and relative read abundance. And relative read abundance, or RRA, is calculated by summing all of the sequences that come from a certain diet item in a scat and dividing it by the total number of sequences in that scat. So in this mock example, let's say we have a scat with 6 sequences, 3 that assign to this lizard and 2 that assign to this bird. 3 divided by 5 is 0.6, 2 divided by 5 is 0.4 for the bird. And so over 2 years, in 2020 and 2021, we collected 1,258 scats, which were collected by myself and several community scientists that are shown here. I wouldn't be able to do this without their help. And so just to dive into the results, this donut plot shows the percent of occurrence of 6 major functional categories in the population's diet as a whole. We have anthropogenic food, marine resources, birds, medium-sized mammals, herps, and small mammals. And to walk you through this figure, each ring represents a different taxonomic classification. So those broad categories are shown in the innermost ring, but as we move, here we have orders, families, genus, and species level designations in the outermost ring. And one of the things that immediately stood out to me is just the large amount of anthropogenic food in the diet. But also of note was this guy, this whale, which was a surprise for me. This is a fin whale. And I was initially confused by the presence of these fin whale reeds in a handful of these samples. And what I noticed is that all of these samples were collected in May 2021 at the San Francisco golf course. For those of you who are familiar with San Francisco, this is a golf course on the coast right by Fort Funston. And as I found myself doing several times throughout this study, I turned to INAT to help me figure out what was going on. And I saw that a den fin whale had washed up on the shore just a couple weeks before these samples were collected. And this was really cool because not only did it really provide this nice confirmation that this methodological approach was working really well, but it's a really good example of how community science data can be really, really informative and useful for eDNA studies. Next, I wanted to look at differences in diet in different territories across the landscape. And so each ring here is a different coyote territory, and we determine these territories using observational data, collar data, and genetic information. And each ring shows the average relative breed abundance of those different functional groups in these different sites. And what is immediately apparent is there is a large amount of variation across these territories. And so I want to focus in on two territories in particular, the Presidio and Coit Tower, which despite being very geographically close by, have dramatically different diets. So the Presidio, shown here on the left, is the largest park in San Francisco. And when we classify the land cover in a 1 kilometer buffer surrounding the center of the territory, only 30% of that land is classified as urban. But when we compare it to Coit Tower, which is a tourist attraction, 98% of the land in that 1 kilometer buffer is classified as urban. This is using National Land Cover Database classifications. And so looking at the species level to figure out what's going on here, it becomes clear what is driving these differences. So these are stacked bar plots. And what I'm showing you is the top 10 diet items for the whole population. And that other category are all the diet items that didn't make that top 10 list. And what we see in the Presidio is pocket gophers make up a really large part of the diet. But when we compare that to the coyotes at Coit Tower, they're eating very, very few pocket gophers. And that difference is being made up by chicken, which is an anthropogenic food that can be sourced from human food, garbage, pet food, as well as direct hand feeding and food that's left for the animals. And interestingly, pigeons and doves, which were not very commonly eaten in any of the other territories in the study. And although the Presidio and Coit Tower are at the extremes of the most urban site and the least urban site, when we look at the correlation between the percent urban cover in one of these territories and the average amount of human food in their diet, we see that it's pretty clear that the most urban sites are consuming the most anthropogenic food. And this makes sense given what I've seen with 2 years of on-the-ground field work in San Francisco. People are hand feeding or leaving food out for these animals at the most urban sites. So these pictures, which I took from the Bernalwood neighborhood Facebook group, are examples of food that's left out nearly daily for these animals at Bernal Hill. And this video is an example of someone feeding a coyote at Bernal Hill. And it's kind of a long video, so I'm not going to show you all of it, but if you're interested about this, I'm happy to show you the rest of it and we can talk about it more. And so looking at individual level diets, each bar here is a different individual coyote. We also see large amounts of dietary variation. For example, these coyotes shown here in the black stars are not consuming a lot of anthropogenic food, but when we compared them to these 3 coyotes in the white stars, they're eating nearly 3 times as much anthropogenic food as their counterparts. And so even though San Francisco is really small, it's really dense, coyotes are generalists, we see a huge amount of variation across the city. However, when we look within a territory, which are shown in these black boxes here, diets are pretty consistent. And they show these similarities within these family groups, within these locations. And this is also not unexpected because coyotes are very social animals. They have high levels of parental care. And so it's not that surprising that pups who learn how to forage and hunt from their parents might develop similar diets and preferences to them. So ultimately, individual variation in the city does exist. And in some cases, individuals develop unique preferences. For example, almost all of the skunk breeds in the entire study were traced back to one individual, coyote number 17, which is shown in the plot on the left. That top green bar is the amount of skunk in this one coyote's diet. However, despite these intricacies, landscape differences really appear to be these key drivers of diet. And coyotes in more urbanized areas are consuming more anthropogenic food compared to those in the less urbanized areas. And what these results really point to is that we, as the people that design and build cities, we have this enormous power because we dictate where resources are on the landscape. And that means we're heavily influencing what urban animals are and aren't eating. And because we design the environment and we build it in this way, it means that we also have enormous power to shape the ecologies of these animals, sometimes in ways that exacerbate conflict, but in other ways that can maximize coexistence if we want to. And so with that, I would like to thank many, many people, my colleagues and interns at the UC Davis Mammalian Ecology and Conservation Unit, my colleagues in the Shell Lab, community members in San Francisco who assisted with sample contributions and many other parts of this project, my community partners in San Francisco, mentors, as well as funders, and I would be happy to take any questions. Yeah.
The joke is nobody knows what's in a hot dog.
Uh-huh.
So what is your search?
All right. Thank you so much, everybody, for being here. I'm really excited to tell you about some new research that we have going on in the Shell Lab, where we're looking at risk-taking behavior and problem-solving ability of carnivores across diverse neighborhoods in East Bay, California. Maybe. Bear with me just one sec. Maureen or okay. No, well, since it's in this, can I just do it like that? It's kind of in this setting, so it's not exactly. Sorry, folks, bear with us.
Yeah, I think the arrow keys work.
It's just okay. Okay, perfect. All right. So cities are complex environments characterized by a mosaic of different natural and artificial elements. And chances are, if you're walking across any city, and as we saw in Tali's presentation, just before this, it's likely that as you're cruising around, you're going to run into some pretty stark environmental differences as you're moving from neighborhood to neighborhood. And so this high heterogeneity that we see within cities largely stems from inequities within our society. So biases in urban planning, as well as historical policies like redlining, have led to an unequal distribution of environmental benefits and stressors across the landscape. And we know that this can be pretty significant for wildlife. There's more and more research coming out that's illustrating how the systemic biases within our society can actually influence the, sorry about this. It doesn't seem to be moving on my end. So yeah, so it's basically influencing these ecological and evolutionary patterns and processes that we can see today in urban environments. And one of the most widely studied aspects of this to date is the luxury effect. So that's where we see a relationship between biodiversity score and, I'm real sorry, y'all. I don't know why it's just not keeping up here. Now I have no arrows. It just kind of stopped.
I think that's the way these people set it up. And
Okay.
yeah, it could be that. There you go.
It just seems to be like locking us. I'm like,
I think it's because you have two screens, like this is one and then there's like another one here.
okay.
Maybe you can't see it. It's the mouse. I think there you go. So because it's across two screens, I think that's like the problem. You need to make sure that the mouse is so that nice here and now another minute. It should. There you
For
go.
me, it doesn't. Okay.
No, it's because it's on two screens.
Okay.
So just
Let me
make sure
try
the
again.
mouse is over there so that it
The
will
mouse
change
goes there.
there.
Okay.
It's
All right,
gonna be
cool.
here in the screen.
All right. Well, thank you. Thanks for bearing with me, y'all. So where are we? Okay. So the luxury effect's probably been the most widely studied aspect of this. And so in this paper here, you can see the authors were interested in looking at mammalian species richness and as it relates to neighborhood wealth. And you can, looking across these different cities, we can see that the strength of the luxury effect does vary city to city. So we know that wealth doesn't explain everything, and we know that it doesn't just end with biodiversity. We know that these differences can actually influence the health and well-being of both people and animals across different neighborhoods. And so here, for example, you can see a map of Washington, D.C., where we are today. And in the map on the left-hand side, you can see that there is an unequal distribution of tree canopy cover across the city. And if you look, and it's the map on the right-hand side, you can see that 16th Street runs up, between the city and seems to serve as this line of racial segregation, where we see more tree canopy cover to the west side of the city in the white, wealthier neighborhoods. And so you can imagine for folks living in the west side of the city, there might be some, health disparities associated with this, primarily within the forms of urban heat islands. So that lack of tree canopy cover and increased impervious surfaces can lead to hotter temperatures. And so on any given hot day in Washington, D.C., we might expect to find as much as a 20-degree difference between these different neighborhoods. And so folks that are living on the east side of the city are going to be susceptible not just to heat stroke and heat exhaustion, but also respiratory diseases like asthma. And another really important health disparity that's associated with this is the distribution of environmental contaminants or pollution across the city. And so what you're looking at here is a map of East Bay, California, where we do our research. And it's a map of pollution burden. And so these pollution burden scores were taken from the Cal EnviroScreen scoring system, which calculates a number of different pollution metrics at the census tract level. And so the darker colors indicate higher pollution burden scores. And we know that for folks living in these more heavily polluted environments, there are a number of health consequences of this, especially related to physical and cognitive development and processing. But what we still don't understand is really how this influences urban wildlife, especially urban carnivores. But based on what we do know with regards to how environmental contamination affects cognition and behavior, we can expect this to be pretty significant. So for instance, risk-taking behavior is really important for animals because it allows them to strike this balance between acquiring resources and avoiding sources of mortality. And there's been a lot of work done in aquatic species where we know that pesticides can negatively influence cognition. So we see altered risk perception and we see bolder behaviors occurring in species like damselflies and perch. And then similarly, there's more research coming out looking at the effects of heavy metal pollutants on songbird risk-taking perception and behavior. And we're seeing an increase in aggression, exploration, as well as bold behavior in some species. And then similarly, with problem-solving ability, this is a really important cognitive skill where animals are, using the skill in order to overcome challenges or solve or, achieve goals, especially within like foraging contexts. And so we know that heavy metal pollutants can negatively influence learning and memory in honeybees, and that rats that are conceived and reared near roadway pollution have delayed psychomotor development as well as demonstrate abnormal behaviors. And then secondary exposure to anticoagulant rodenticides, which urban carnivores are especially at high risk of, can lead to prenatal development issues as well as a lot of just organ and immune function issues. So you might be wondering, well, how exactly does this work? How does environmental contamination influence the brain and behavior? And the answer is there's a lot of different pathways by which it might operate. One such pathway could be oxidative stress. So when animals are in a stressed environment or exposed to pollutants, we see an abundance of free radicals, and it creates this imbalance with antioxidants, which leads to tissue degradation and to cognitive degradation. And this could be really important for certain behaviors such as foraging and risk-taking behavior, and especially for mesocarnivores that are oftentimes in conflict with humans. So over the past few years in the Shell Lab, we've been working to better understand how societal inequities within the East Bay area could be spilling over to jeopardize the well-being and health and behavior of urban carnivores. So as you can see, this is a map of our UN transects in East Bay. The base map here is poverty levels. So the darker colors indicate higher levels of poverty. So we have one transect that runs through a lower income communities, and then another transect that runs through higher income communities. And so for the past 6 months or so, I've been leading an experiment where we've been repurposing these camera trap locations and expanding upon them in order to conduct behavioral and cognitive assays of carnivores. And so we're doing this by partnering with many local organizations, including the Oakland Zoo, East Bay Regional Park District, and a couple of local nonprofits that are focused on bringing outdoor education and experiences to students in underserved communities. And the way this works is that we're setting up, a camera trap in folks' backyards and in other green spaces, and we're placing a puzzle in front of the camera. And so the puzzle is this black object on the ground. It's anchored to the ground using a lag screw, and inside of it contains a very small cat treat. And so the animal has to figure out how to open the tube in order to get the cat treat out by clawing at the end of the cap or pulling on the rope. And so with this, we are asking 3 primary questions. First, we are just looking for patterns of different carnivore species across neighborhoods, what neighborhood features might be influencing the distribution of different carnivores. Second, we're asking about the risk-taking behavior. So are they willing to even interact with this puzzle? And then finally, if they do interact with it, are they able to figure out how to open it in order to retrieve that cat treat? All right, and so these are 3 photos of yards from different neighborhoods that we work in. So the one to the far left is mostly impervious surfaces, and any vegetation that you see there is in pots. And then the middle panel is more along the lines of most of our current sites to date. So it's a small yard with a fence. It has permeability, so we know animals are coming in and out of there, and just varying amounts of vegetation. And then to the far right, we have some other sites which are larger properties. They're not fenced in. They're adjacent to canyons and other open spaces. So we know that carnivores are moving through these areas as well. And so this is a series of video clips from our most common species that are, coming into contact with the puzzles and participating in our experiment. So first up, you're going to see a possum who's fighting at the tube. Here's a striped skunk spinning the tube around. Here's the domestic cat. So we are getting a lot of cats. I mean, surprise, surprise, they're interested in the cat treat inside. This is a gray fox.
Yeah.
And here we have coyotes. So this is a coyote in somebody's backyard. Here we have a coyote that's in a cemetery. And it may or may not surprise you, but our #1 repeat customer who seems really into this experiment are raccoons. So raccoons are really interested in the puzzle, and they're absolutely killing it. You can see here we have a raccoon that just opened the tube and another raccoon that just solved the puzzle. All right, so as I said, this is a new experiment, so we're still running testing, but I do have some preliminary results that I'd like to share with you. So to date, we've tested 57 different locations. At 33 of these sites, the puzzles have been solved, but at 24, they have not been solved. And we have 3 wonderful undergraduate students that have been helping us with this research. So they've been going through all of our trail camera videos and going through the footage. And so based on the data they've collected so far, I went through the first observation of each species at each location and categorized what the response to the puzzle was. So in the top color, I believe it's pink, and the color at the top of the bar, you're going to see individuals that did not interact with the puzzle. So for the time being, we're considering these individuals to be risk adverse. And then the next block of color, those are going to be individuals that did interact with the puzzle, but they were unable to solve it. And then at the bottom of the graph, you're going to see the individuals that did solve the puzzle. So they were successful in opening it to get out that cat treat. And so here, each bar in this bar chart has our different species. So you can see, on the farthest to the left, we have raccoons. So about 1/4 of the raccoons are not interacting with the tube. 1/4 are interacting and they're not solving it. But about half of them are interacting with the tube and they are solving it. And then if we move on to possums and skunks, you can see that the majority of individuals are approaching and interacting with the puzzle, but they're having lower success at opening the tube. And then if we move on to coyotes and domestic cats, we're seeing less engagement with the puzzle, and we're seeing less problem-solving success. But now if we move on to just ultimately, who solved the puzzle at what location, you can see each square on this map. Again, this is our pollution burden score map. You can see that each square is a different camera trap location. So the open squares are places where the puzzle was not solved, and the filled in squares are places where it was solved, and each color corresponds to the species that opened it. So again, raccoons, who are the gold squares, are doing really well. They're opening puzzles all across the place. And we do have a couple of coyotes that have solved and a couple of opossums and skunks as well. And again, this is very preliminary, but at the moment, we are seeing this trend towards more puzzles being opened in neighborhoods with lower environmental contamination, whereas our puzzles that are being unopened are in areas of higher environmental contamination. And this is what we would expect to see if there are some effects of pollution burden on cognitive abilities. All right, so in sum, we have found that we're seeing higher risk-taking behavior in raccoons, possums, and skunks. This is that makes sense to me because, we know that these 3 species really do use a variety of different anthropogenic resources. They'll feed on garbage, they'll den in people's homes. So we might expect to see greater risk-taking behavior in these species. And again, raccoons are the most successful at this task to date. And I think there's a couple of reasons why this is the case. So, we know raccoons are really good problem solvers. Raccoons also have really impressive brains. So the neuronal density in raccoon brains is actually more similar to that of primates rather than other carnivore species. And in addition, they're really great extractive foragers. They're really good at opening things in order to get food. And these two traits are not unrelated. So we actually do think that one of the hypotheses for the evolution of intelligence is of extractive foraging on environmental complexity. So, with regards to our the distribution of the problem solving that we're seeing, we are seeing greater problem solving success in these areas of less environmental contamination, but at the moment we don't know why that is the case. So we are, as far as our next steps go, we're going to be repeating this experiment later this fall and into the spring of 2024, and we're going to be moving into other neighborhoods with higher pollution burden. So you can see on our map here, these darker colors, we're going to increase our sampling in those areas. And then in addition, we're going to be quantifying a number of different additional variables that could be influencing our results. So, some of the things we're going to be looking at is environmental complexity, small scale features of different yards. And then we're also going to be doing surveys on human attitudes to see how that might be influencing species presence and their willingness to engage, with our puzzles and participate in this study. And then this is going to allow us to kind of create like a map or get an understanding of the landscape of different cognitive challenges that are out there for them, risks, as well as attractants and deterrents, and see how that might be playing a role. We're going to continue some more coding of the videos. We're going to be looking for more fine scale aspects of cognition and behavior. So we're going to be looking at exploratory diversity, which is the number of unique behaviors that the animals direct towards the puzzle. We know that this predicts problem solving success. We're also going to look at dexterity, and both of these things are going to allow us to evaluate behavioral flexibility as well as psychomotor skills and see what could be happening there. And then finally, we are slotted later this year to begin carcass collections as well as trapping of mesocarnivores in East Bay. So this is going to allow us to collect a number of biological samples that will tell us about the overall health of the animals, their stress levels. We're also going to do behavioral assessments to measure aggression towards human handlers and docility, and start to understand more of these, attributes that could be, influencing health and stress and creating a causal link between environmental contamination and cognition and behavior. Okay. Ooh. And so with that, I would like to thank you all so much for being here and for bearing with me as we had to, figure things out. I am so grateful to all of the partners that have worked with us on this project, as well as, all of the residents that have just been really jazzed to be participating. They're just as excited, if not more excited than me, to check out the video footage and see, who's in their yard and what they're doing. I also want to thank my amazing co-authors on this work, Chris, Cesar, and Christine, all three of whom are going to be talking in the symposium on Wednesday on One Health in Wild Cities. So I strongly encourage you to check that out. And I think if there's time for questions, I'd be happy to take them now. Okay.
Are you putting out these,
Yeah.
One,
yeah, right now, one.
you don't have anybody learning how to.
No,
do
just
these
one
people
time.
do?
We have, we do plan against some crowd and marketing for animals, which would allow us, if we wanted to look across different observations. We know that's the range of animals right now. It's just a one-time thing, one and done, because we don't know.
How do I move between? How do I move between slides? Okay, so you're gonna, because she changed it, and I don't know how she has it. It's good now. So you make sure this is one screen and there's another one here. So the mouse has to be there, like not here.
So I won't be
There.
able to see my.
So then,
Slides.
because that's how she set it up, I'm so sorry. And then you move it, just make sure the mouse is on here. I'm so sorry.
Okay. Hi, I'm Jasmine Grewal. I'm a master's student at the University of Nebraska-Lincoln, and I'd like to acknowledge my co-authors, Justin Brown and Seth Riley with the National Park Service, and my advisor, John Benson at UNL. And today I'm going to be presenting our research on coyote space use in Greater Los Angeles. So when we think about how animals use space, it's often tied to the availability of resources on the landscape. The optimal foraging theory predicts that animals will try to minimize the energy they expend while meeting their resource needs. So this often means a trade-off between minimizing home range size and maximizing the resources they're able to acquire. So for an animal that has an abundance of resources on the landscape, they don't have to go very far, so their home range size can stay small. In other areas where resources are more dispersed, animals have to move farther to find food, so home range size often increases. Aside from resources, risks can also play a role in influencing home range size. So animals that have predators or competitors on the landscape will avoid those negative interactions, also increasing home range size. And according to the risk disturbance hypothesis, this anti-predator behavior can also be displayed towards human disturbance, even if that disturbance is non-lethal. So in areas with humans, home range size often increases as well. On the urban landscape, physical changes to the landscape also impose risks. So fragmentation from infrastructure like roads and buildings leads to animals having to move between natural patches to find food, and that also can increase home range size. However, in urban environments, there are also benefits for species that are able to take advantage of them. There's an abundance of anthropogenic food subsidies coming in the form of human food waste, ornamental fruiting plants, and synanthropic species, which are animals that thrive in urban systems like cats, house mice, and house sparrows. However, not all animals are able to equally take advantage of these resources. Mountain lions, for example, are obligate carnivores, so they can't take advantage of many of the things like human food waste in these systems. However, there might be an increase in their prey species in urban environments. In a study in the Greater Los Angeles area on mountain lion home range sizes, it was found that home range size increased with the level of development. However, at the most extreme levels of development, such as in downtown Los Angeles, they found that the home range size had a sharp drop off. So that's where you find P22 in Griffith Park with one of the smallest home range sizes studied in mountain lions. And this is because they have larger space use needs. And at that very extreme level, the fragmentation becomes a barrier for them. On the other hand, animals like raccoons are able to take advantage of pretty much all of the food that can be found in urban areas, and they have smaller home ranges in urban areas than in rural areas, and aren't as negatively impacted by things like fragmentation. But they need to be able to access these resources without encountering humans. In a study in Yellowstone, it was found that elk were accessing risky habitat at safer times by using that habitat at times when wolf activity was lower. In a similar way, urban animals will shift their activity patterns temporally towards nocturnality to avoid human encounters while still taking advantage of those anthropogenic foods in cities. Urban coyotes are particularly interesting to study because they kind of fall in the middle of this spectrum of animals that are negatively impacted by urban systems due to fragmentation and because they're seen as a conflict species in many instances, but they also are able to take advantage of the food sources in cities. It has been found in some areas that that home range sizes increase with increasing development. In other areas, it's found that home range sizes for coyotes decrease with levels of development, and some areas have found no relationship. We are specifically looking at the greater Los Angeles area. So greater Los Angeles is home to almost 13 million people. It's a very highly developed downtown area surrounded by urban and suburban sprawl. At the northern end, we have agricultural land, and woven throughout, we have this matrix of natural cover, largely from the Santa Monica Mountains National Recreation Area, as well as state and local parks. And we were able to track coyotes in all of these different land covers. And previous research in greater Los Angeles, they have studied coyote home range sizes, but it was mainly focused in these kind of suburban areas near the Santa Monica Mountains. They did find an increase in home range size with increasing level of development. And this data was also taken using VHF telemetry, where we will be using GPS telemetry. In addition, a coyote diet study in the greater Los Angeles area found that in the most highly urbanized areas, the diets of coyotes were more reliant on anthropogenic food subsidies, and they were more homogeneous. Whereas in intermediate levels of development, where you have more of this matrix of different cover types, the variation in diet was greater within individuals and between individuals. So when I'm talking about the different levels of development, it's important that we have a definition for what that means. And we took our definitions from the National Land Cover Database. So open development is altered open land, which includes lawns, cemeteries, golf courses. Low intensity development are areas with single family homes that are more dispersed. Medium intensity development has single family homes at higher densities, and high intensity development are things like downtown areas with industrial areas and more development in apartment blocks. We are considering 3 hypotheses to look at coyote space use. The first is in relation to home range size. The second is in relation to resource selection within home ranges. And the third is with temporal variation. Our first hypothesis is that in landscapes with high levels of risk and reward associated with humans, increasing heterogeneity leads to variation in space use by generalist species. So this means in areas where there are more different land cover types, animals are able to specialize differently within that. Our first prediction is that suburban coyotes will have larger and more variable home ranges. And our second prediction is that in urban areas, the home ranges will be smaller as there are more abundant resources and also higher risks associated with moving further out of the area. We are using data from 44 collared coyotes that were collared between 2012 and 2022. We excluded 1 coyote that dispersed during the study period. So we think that all of the coyotes studied are resident coyotes. In total, we had about 52,000 GPS fixes. So we use these to estimate home ranges using adaptive local convex hulls. And then we estimated percent cover within home ranges using the National Land Cover Database and those different levels of development that I just discussed before. So we created models. The first 2 models show the different levels of development by percent cover, in addition to natural cover. And then we also considered models using the density of roads within home ranges. We use a Kaike information criteria to rank the models, and the lower the AIC value, the better fit the model was. As you can see here, the null model with no variables was the lowest AIC value. So none of our models were supported. So we didn't find a relationship with the percent cover and the home range size of coyotes. We then categorized the coyote home ranges into larger scale context land use. So we had 4 categories, including agricultural, natural, suburban, and urban. Our natural home ranges were 10 of the coyote home ranges, and they were predominantly undeveloped land. Agricultural home ranges were areas that were mainly cultivated crop and pasture land. Suburban coyotes, which was our largest sample size, was predominantly developed land at less than 45% constructed materials. And our urban coyotes, which you can see here, we're in downtown Los Angeles, were predominantly medium and high development at greater than 45% constructed materials. We then found the average home range size for each of these categories, and here they're plotted with standard error, but if you look at the confidence intervals, they do all overlap, so we didn't find a significant difference with home range size between these land use types, but you can see that there is a larger variation in home range size of suburban coyotes. And to exemplify this, here are two home ranges from coyotes. One is about 100 kilometers squared, and the other is only 1.5 kilometers squared. And these are both suburban coyotes that have a large proportion of natural area around the suburbs going through the middle. But you can see that they specialize differently. They have different strategies to obtain resources and very different home range sizes. To look at resource selection, we looked at resource selection within home ranges, and we predicted that the resource selection of coyotes is variable dependent on the broader landscape context of the coyotes' home ranges. We first predicted that urban and suburban coyotes would show a relaxed avoidance of high and medium development, as it is more commonly found in those home ranges. That natural coyotes in natural areas would display a stronger selection of development because those are less represented in those home ranges, but also represent high reward because of anthropogenic food subsidies. And that coyotes in agricultural areas would avoid low development more strongly because in these rural areas, humans may pose a greater risk due to perceived conflicts. To look at third order selection, we compared used and available resources. So within the home ranges, we used the GPS telemetry locations from our collars and compare that to the National Land Cover database cover types that were available within the home ranges. So we ran a resource selection function, and I know this is a lot of data on one figure, but I'll walk us through it. So the y-axis shows distance to these cover types. So any negative values show selection to those cover types, and positive values show avoidance for the beta coefficients. Looking at high development, coyotes in natural and agricultural areas did select the high development cover types more strongly relative to their availability. And again, this is likely due to the increased availability of food sources in these areas. There was avoidance of medium development. At low development and open development, we begin to see urban and suburban coyotes showing selection to these cover types, and that is likely because medium and high development are found more in these home ranges, so they are moving to open and low development to avoid interactions with humans while still taking advantage of anthropogenic food subsidies. Oh gosh, how do I go back?
Oh,
It went to the last slide.
You've been signed out because you're currently signed in on another device. That I don't know because, okay, that's gotta do with the Zoom thing. I will tell you how to do your slide. Okay. Let's see here. You were here in this one?
Backward.
This is the beta coefficient. Hypothesis 3.
Yeah,
This one, hypothesis 2.
yeah, I think we're there.
This one, right? Okay. So just keep using the arrows.
And then interestingly, looking at grassland and shrub cover, which were the dominant natural cover types, there were different responses with across all land use types. Coyotes were selecting shrub, but in suburban, natural, and agricultural, they were avoiding grassland and herbaceous. Our third prediction has to do with temporal variation in resource selection. So we believe that in these areas of higher urbanization, animals would be shifting their activity patterns nocturnally to access resources when humans were not present. So we expected higher resource selection of medium and high development at nighttime. So looking at high development, we see that there isn't a large difference between, okay, so the darker dots on this plot are showing nighttime resource selection, and the lighter ones are showing daytime resource selection. So we can see at high levels of development, there isn't a large difference between day and night. And this is likely due to continued risk throughout day and night for coyotes. At medium levels of development, we begin to see a shift of relaxed avoidance during the night. And that's likely because there's less human activity at these times. And then at low and open development at day, we see stronger selection in urban coyotes, and this is likely because they are avoiding medium and high development during the day because of human activity. So they move to areas of low and open development at those times. So to summarize, we did not find a relationship with home range size and percent cover of different development types, but we did find that at those intermediate levels of development where the landscape was more heterogeneous, there was an increase in the variation and range of home range sizes. In terms of resource selection, we found that they did differ in how they were selecting resources relative to how they were available, depending on land use type. And coyotes were shifting their resource selection, especially in the more urbanized areas, relative to human activity. And this study helps us to see how an animal like coyotes that are highly adaptable are able to specialize across an urbanization gradient to take advantage of human food resources while also avoiding risks from human encounters. And I'd like to thank our collaborators, the technicians and biologists at the National Park Service, my lab mates at the Benson Lab, and the School of Natural Resources at the University of Nebraska-Lincoln, as well as California Department of Transportation for funding many of the projects that led to this data set. Any questions?
I'm curious if you look at sex differences. We have not. That is something that we're going to be looking at, but previous studies haven't found large differences between home range size and sex. But that is something that we will.
If it's gonna cause problems, I'll just cut my losses.
I'm gonna just go with what I know.
Yeah, that's
Sorry.
cool. No worries.
Is that cool?
Yeah. if we could-
Oh, we can drag up your notes.
And I don't want the notes, I just wanted it nice. Rolling.
Well, let's play with it. Oh, maybe we can like,
Okay, I mean, the way you had it before we started. Well, that's.
full screen over here.
All right, our last speaker is John Benson from the University of Nebraska at Lincoln.
But-
Who is gonna mess with some maybes?
But can't we make it? I mean, I don't know how I'm gonna,
And she just sets it in a way that it
it's not gonna advance though, if it's like this, you know.
let me see if I can fix it for you.
Thank you.
I can't promise anything because she set it up in a way that it just doesn't make much sense. Let's see. Could you do that? No.
Maureen, the way you had it initially would be great, because right now it won't advance in this mode. And so we just, whether it's notes or no notes, I don't care, but it just as long as it's in a presentation mode, that would be good.
That doesn't advance the other one. You have to set it up. Right.
Okay. Okay, let's see here.
You have to duplicate it that are,
Can you come here and say that again, please? What are you saying?
you have to duplicate it. So-
Can you do that?
Let's see if I can.
Yeah.
So this is here. This is here. Okay, so now. If we go,
Sunshine.
where is? Here.
Oh,
Okay, so
yeah,
do it like that.
Great.
Let's just.
Second.
That's better, yeah, that will
Yeah.
work.
Okay.
Thank you.
got it.
Thanks, Gavin. Okay, thanks everyone, and thanks for your patience. So great to be with you here today. I'd just like to start by acknowledging my co-authors. So Kyle Doherty is a PhD student in my lab at the University of Nebraska, and Jeff Sigich and Seth Riley have led the research with the National Park Service for these mountain lions for over 20 years. So we know that animals make decisions about risks and rewards as they move through landscapes. And we've learned a lot about forage predation risk trade-offs as prey species try to get enough to eat without being eaten themselves. But we're increasingly recognizing that predators and other animals may respond to human disturbance in a similar way that prey species respond to predation risk. And Frid and Dill articulated the risk disturbance hypothesis, suggesting that there may be behaviorally mediated indirect costs for predators and other animals as they respond to human disturbance. So large carnivores are balancing energy and human risk as they move through landscapes. But I think there may be another source of risk that's been largely ignored. And so these are big, dangerous animals, right? And they do kill each other. And so potentially they may also, as they're navigating landscapes, they're thinking about human risk, they're thinking about food, but they're probably thinking about each other as well. So this research that's been going on for over 20 years in Greater Los Angeles is centered around a small population in the Santa Monica Mountains. And this is a highly isolated population, isolated by large anthropogenic barriers, major freeways. There's high rates of inbreeding and very low genetic diversity. And we built some models that actually predict local extinction risk from a combination of demographic and genetic risk factors in the next 50 years. And the leading causes of mortality are collisions with vehicles and also interspecific killing, so lions killing other lions. And we think that may be exacerbated given the space limitations and these anthropogenic barriers. But regardless, we've documented adult males killing kittens, subadults, which would be independent age, independent from their mothers, but before they breed themselves, so subadult lions, and then also even adult females. Mule deer are the primary prey for mountain lions in this system. And in a paper in 2016, we found sex differences in where they kill mule deer across Greater Los Angeles. And so in our resource selection function at kill sites, we found that males selected riparian woodlands more than females, whereas females selected development more than males. And that led us to hypothesize that maybe females are avoiding the best natural landscape features when killing deer and avoiding those features to avoid dangerous encounters with males who are using those areas. And then they may compensate by actually killing deer closer to development. And so this leads us to our hypothesis for today, and that's that interspecific risk may alter the trade-offs between foraging success and human disturbance for these lions in space-limited environments. And so our first prediction is that adult males will select natural landscape features associated with dense stalking cover, which should lead to higher predation success. We second predicted that female and subadult selection of foraging areas is modified by trying to avoid encounters with these dangerous males. And we actually looked at proactive and reactive avoidance, and I'll talk about what that means in a second. And third, our third prediction deals with time. And so our prediction here was that all lions will relax avoidance of human features at night relative to crepuscular features, but that these temporal shifts would be weakest in the adult males, who again are probably able to prioritize the best natural landscape features. Okay, so we had GPS collar data from 49 mountain lions, and these collars get locations every 2 hours from 5 p.m. to 5 a.m. So our movement models are based only on crepuscular and nighttime data, no daytime data. We started with a third order resource selection function, so within home range resource selection function for adult males, and that gave us a probability surface for where animals will be most likely to encounter these males. And so we consider this to be potential proactive responses. In other words, do subadults and adult females avoid or select areas where they're most likely to run into these males? In terms of reactive responses, we look directly at the GPS telemetry data from the nearest adult male. And so when these subadults and females are moving around the landscape, if an adult male came within a certain distance and we set that distance at 5 kilometers, how did the animals respond? Did they move away? Did they move towards the male? What did they do? So that would be the reactive response. And most of this is based on step selection functions. So we use these to evaluate movement-based resource selection and directly quantify movement decisions between telemetry locations. And it just jumped to the end, but I'm gonna try to make this work. All right, we're back. Cool. Okay, so you can imagine you might have a series of sequential telemetry locations on mountain lines separated by 2 hours, as we did. And so here they are. We could measure the empirical step lengths between those animals, but we could also measure the turning angles along the movement path. And the combination of these empirical step lengths and turning angles, the movement behavior of each individual, allowed us to set up a series of available steps. These are steps not taken by the lion, but steps they might have plausibly taken given their previous movement behavior. And at the end of these steps, we can set available locations, and we compare landscape features at the available locations to landscape features at the steps the animals actually took to derive some information about how animals make decisions as they move through the landscape. Okay, so starting with the kill site RSF. So this is a resource selection function where the used locations are sites where mountain lions killed mule deer, and the available locations were distributed across their home range. This is essentially us redoing our 2016 paper with more data. We now have 515 kills from 33 mountain lions. And I want to orient you to this. So similar to Jasmine's talk, we've got a lot of distance-based features. All the natural and anthropogenic landscape features are distance-based variables. And so it's a little counterintuitive. You can see the red line there. Beta coefficients below 0 or below the red line are actually selection. Above the red line would be avoidance. And what you can see is the natural landscape features with stalking cover. So the shrub and woodland habitats were selected at kill sites. In Chaparral, you can see a dense shrubby Mediterranean habitat in Southern California was one of the stronger responses where animals are selecting these habitats when killing mule deer. We moved to prairie meadows. So this is another natural landscape feature, but without that dense stalking cover, and they actually avoid those with killing mule deer. Altered open, this is basically areas disturbed by humans, but not actual developments. We're talking about graveyards, cemeteries. Graveyards and cemeteries are the same thing. Golf courses and like manicured parks, that kind of thing, lawns. And then next we had, so they basically showed no response there, a trend towards avoidance. They actually selected development strongly when killing deer. Now, remember, this is distance-based selection, so they're not actually killing deer in backyards typically. In fact, after 515 kills, I think we had 2 or 3 that were within development. And one thing that people sometimes forget to do, it's really important to look at, well, where actually were the animals? So we're seeing strong selection for development at kill sites, but on average, those kill sites are actually 1.5 kilometers from development. And this is actually a cool thing about distance-based variables, is you can pick up selection for features that the animals are actually rarely in. And we can contrast that with Chaparral. So the 2 strongest responses are selection of Chaparral and selection of development. Very far from development, even though they're selecting it, they're just closer on average. But Chaparral, they're really close, and a lot of the kill sites fell within Chaparral. And again, it has that dense stalking cover. Elevation and slope are not distance-based variables, so they're what you'd expect. If the coefficients are below the red line, below 0, then they're actually avoidance. So basically, mountain lions are killing deer at lower elevations and on gentler slopes. Okay, so let's revisit our first prediction. And that was that males will select natural features associated with high predation success. And so we had partial support. Males did select natural features with that good dense stalking cover at kills. But unlike our 2016 paper, we actually had weak support for differences between the sexes at kill sites. The important thing that comes out of here is a probability surface for where lions have the highest probability for killing mule deer in this landscape. And so we can input that into our other models to see if they're selecting those areas where they're most likely to be able to kill deer. Okay, these are a series of slides about the step selection functions. So starting with the adult males, data from 17 males, similar to what you've seen before. So these are distance-based variables here, the landscape features. And we can again see that they're selecting the shrub and the forested habitats with that dense stalking cover along their movement paths, the adult males are. And again, we see they're avoiding prairie meadow where there is no dense stalking cover. The anthropogenic features, no strong selection or avoidance for altered open or development, but we do see that they're avoiding both road types, primary roads, which are major freeways, and then secondary roads, which are also large roads, but not as large as the major freeways. Again, we see basically avoidance of elevation and slopes. So they're at lower elevations and gentler slopes. And really importantly here, they are selecting those areas with the highest probability of killing mule deer. Okay, so let's look at the other age and sex classes. So now we have all age and sexes here. We have adult females, we have adult males again, subadult females and subadult males. I realize this is a pretty busy figure, but we'll get through it together. Again, we've got these distance-based variables. And the thing I really want you to just focus on here is those adult males. You can see for 3 of the 4 habitats with that good dense stalking cover, the shrubs and the forests, males are showing the strongest selection for those for those resource variables. Coastal sage scrub would be the one outlier, but they're also selecting that. And then they're actually the only age and sex class that avoids prairie meadow, the one natural landscape feature without stalking cover. Now, here's the most important part of this slide. And that here we see that all age and sex classes, not just males, are selecting the best areas where they can kill deer. And that's contrary to our prediction. Okay, so let's look at those proactive and reactive responses to adult males. And we're going to cut right to the chase here. And so you can see that the adult females and the subadult classes, none of them are avoiding, inconsistent with our prediction, none of them are avoiding proactively adult males. In fact, they're all showing a trend towards selection. Only the subadult males significantly select areas where they're most likely to run into adult males, but they all show a trend towards it. And regardless, none of them avoided it. So our prediction was not supported. What about reactive responses? And again, here we're looking at how do these animals behave, the subadults and the adult females, when a collared adult male comes in to relatively close proximity. And again, we'll cut to the chase. And again, our prediction is not supported. And so here in green, we can see adult females are actually selecting the nearest adult male when they're less than 5 kilometers away. And we see basically no response by the subadults. And so again, no support for our prediction and no reactive avoidance of adult males. So one thing that occurred to us, we see this apparent selection of females for adult males. And we kind of thought, well, is that real or is that maybe the males selecting the females more likely? And so we looked at both. And on these figures across along the y-axis, you basically have that selection coefficient again. So values below the red line are going to be selection. And then across the x-axis, we have increasing distances. So if you look at females first, from about 20 kilometers to 7 kilometers, you see no selection or avoidance. After the male gets within 7 kilometers, we start to see that increasing trend of females selecting that male that's in close proximity. The effect sizes are pretty big, but there's a lot of variation as they get closer and closer up to 100 meters away from that male. Now, if we look at the males, we see a pretty similar thing where up until about 5 or 6 kilometers, there's no response. Then the males start selecting those females, and that makes a lot of sense. You can see the effect size isn't as big. They are selecting them strongly, not as strongly as females appear to be selecting males, but the coefficient, the variation around the coefficients is a lot tighter, perhaps suggesting it's more the males honing in on the females. Honestly, at this point, we're not totally sure who's selecting whom. And so what we're starting to do is look at the turning angles, and it was kind of a mind bender where we're trying to figure out, look at the turning angles and say, well, what's the true bearing to the other animal? And then what's the bearing that they're taking, and look at who's kind of honing in more on the other one. We're not there yet, but I'll come back to that at the end of the talk. Okay, so let's revisit our second prediction, and that's that female and subadult selection for foraging areas is modified by avoiding these dangerous adult males. And this really wasn't supported. So adult females and subadults select the best areas to kill deer. Basically all age and sex classes did. No proactive or reactive avoidance of males. The one thing that does, I guess, provide a shred of support for our prediction is that we did see weaker selection of adult females and the subadults compared to adult males for the habitat types with that good stalking cover. Okay, so our third prediction again was about time. And so here we've got another overwhelming coefficient plot, but again, the age and sex classes are color coded, and now we've got 2 coefficients for each age and sex class of the same color. The lighter one is during crepuscular periods, morning and evening. The darker or solid one is at night. And we won't go through all of them, but altered open is a good one to look at. You can see in green the adult females. They actually avoid altered open during crepuscular periods and then strongly select it at night. And you see that same trend of either relaxed avoidance or even selection as you move from crepuscular to nocturnal periods. And we found this one particularly interesting. So these are subadult males in black and their response to both road types, primary and secondary roads. You can see during crepuscular periods, strong avoidance of these roads, but during the dead of night or during nocturnal periods, they're actually strongly selecting them. So subadult mountain lions are the dispersing age and sex class. Both males and females disperse. All males disperse as subadults and about half the females do. And males make longer and riskier dispersal events. So we think in this landscape that's dominated by humans and there's a lot of roads that they avoid them during crepuscular periods, but at night, they're more likely to approach them and even cross them as they're trying to find ways to disperse and find their own home range. And of course, crepuscular periods would be consistent with rush hour traffic. And so that makes a lot of sense, basically. Okay, so our third prediction that lions would relax avoidance of human features at night relative to crepuscular periods, and that these shifts would be weakest in adult males was mostly supported. Lions did relax avoidance at night, all age and sex classes, and the subadult males and the adult females exhibited the strongest temporal shifts, much stronger than the adult males, lending some more support for this prediction. Okay, so some take-home messages. Our kill site work certainly shows that good predation habitat involves thick stocking cover, which is consistent with other lion populations. But here in Southern California, that Mediterranean chaparral habitat seems particularly important. But they also, again, selected in a distance-based context. They selected development, not in development, but they're closer than you'd expect across the landscape. We found that adult males exhibit the strongest selection for the natural landscape features that had that dense stocking cover. And we saw this interesting shift between crepuscular and nighttime periods around anthropogenic landscape features. And that's pretty cool. You know, in a lot of landscapes, we see that shift from day to night, but in the heavily human dominated landscape of LA with tons of human activity, we see a pretty strong shift even between crepuscular and nocturnal periods. Okay, so what about this potential interest-specific landscape of fear? Certainly not as we predicted. That doesn't mean it's not happening on some level, and there's clearly reason to stay away from those males. But we did find that all lions selected the best areas for killing deer, so they don't seem to be getting bumped away from the best foraging habitat by the danger of males. We found no proactive or reactive avoidance of adult males. And adult males and females appear to be selecting each other. And so why would that be? Well, clearly there's good reason for a male to select a female and approach a female, see if there's a mating opportunity there. In some cases, females might be doing the same thing, right? But the thing to keep in mind is that female mountain lions, adult female mountain lions, spend the bulk of their life raising kittens or being pregnant. And once they are freed from those kittens, either through mortality or dispersal, there's just a very narrow window when actually mating and copulating with a male would be beneficial to them. In our landscape, it turns out it's usually within a week. It's always within a month that they get pregnant again. So it's just a very small fraction of their life that they should actually be selecting those males. So why else would they be moving towards males? Well, it could be just that adult breeders are checking in on each other as they move through the landscape. And we're not exactly sure why they would do that, but we're actually hoping to look more at these interactions in the coming months and try to learn something about that. Another interesting thing is something I observed when I was working with Florida panthers a number of years ago, and we wrote up as a natural history note, was this idea that there may be a pseudo-estrus response. And so what we observed in a couple of cases was an adult female with an active den with young kittens actually moving towards a male, leaving the kittens and consorting with that male for 2 or 3 days, even though she had no reason to be mating with the male. She already has an active den. And so we took that to mean, and actually when they split up the male and female, she beelined back to the den and raised those kittens to adulthood. And so we took that to mean it's a defensive mechanism where they basically approach the male and cut off the male and keep it away from the kittens, sort of give the male maybe what he wants, spend a few days. Once the male moves on, you head back and raise the kittens. It's actually a pretty cool story, maybe for another day. But we were almost forced by agency pressure to remove those kittens. And luckily we didn't. She came back and raised them to adulthood. Okay, anyway, clearly there's a lot more we can learn here about spatial and social interactions about mountain lions. I think it's particularly interesting within these human dominated areas, because again, we know they're paying attention to humans and they have reason to avoid us. We know they need to get a lot to eat, but they also have reasons to stay away from each other at times. So thanks a lot. Just like to acknowledge my funding sources and organizations, and I'll take any questions.
Hi, yeah, I told you had time because we had a gap, but actually the talk has been moved into this room, so we
Alright.
do not have time for questions. Very sorry about that. Because yes, we have a surprise talk from Richard Halberin. That sparks someone left. Oh, here we go.
Gabby Gabby left.
Do you know PowerPoint? I'm having some issues with PowerPoint where I like hit present, but I'm not getting, but it won't advance.
Okay, well, it was advancing a second ago.
Oh, good.
Oh, but this is not advancing.
Yeah, is that okay with you?
That'll be tough. Because it'll be different than, when we play the game.
Sure. Maybe just X out of it and start over. Because I think I got it and then it went away when I went back to Zoom. And excellent.
All right. Thank you all for making that quick pivot. We'll go back to the main room schedule that's in the program after the coffee break. This is just a one talk pivot to give us some more space from the boardroom. So I'll let Richard take away his talk for this session. Thanks, Travis. You guys keep an eye on the time, and when it's time for me to shut up, just Travis, you just come and yeah, maybe Seth can do it. And when Seth stands up, I'll stop talking. Thank you all for having me. Thanks for pivoting to this room. The presentation here is going to be a departure from the great talk we just heard in the other room from Seth. A lot of data, a lot of good science. This is not that, okay? What this is this is the description of a lessons learned effort. We had these big visions in San Antonio where we some of the local Audubon chapters brought in Peter Mara, who wrote with his co-author the great book Cat Wars, and everyone was fired up about we're going to finally do something. We had all these great ideas, and so I want to tell you what we did, why it didn't work, and how you can learn from those mistakes with just a little bit of science mixed in. So I want to, for those of you who haven't thought about this in a while, an overview of some facts, because we're scientists and that's what we do, right? As we learn facts and home ranges and diet studies. Give you the impacts of feral cat depredation, take you through what we were trying to do in San Antonio, evaluate just a very basic ad hoc home range lit review, and then what we produced and then why it didn't work. So I want to tell a story. About 15 years ago, I was at a city council meeting and I was not testifying. I was just observing, and they was talking about trap, neuter, release policies. And the birders got up there and they gave very well-informed, well-reasoned, well-researched presentations about mortality rates and disease transmission and depredation and endangered species and birds and how I like birds and the numbers of jobs that the birds comes in and the impact of hotel and tourism and all this stuff. And it was fantastic. And I was cheering them, and I was nodding my head. And the city council was not nodding their head. They were just politely listening. And then the birders got up. Excuse me, the cat advocates got up. And she didn't talk about science. She didn't talk about depredation rates. She told a story about how she was widowed. Kids are gone, fixed income, friends are gone, no neighbors, how much she loves the cats, how much she feeds the cats, how much the cats depend on her, emotional connection, how much joy the cats bring to her life. And then she started listing the names. And of course, I'm very professional, but in my head, I am rolling my eyes so far in the back of my head, they hurt, right? But guess who was nodding? The city council was nodding, right? She had established, she and all the other cat advocates that came after her had established an emotional connection. She had told a story and that connected with the audience. Now, truthfully, even if they hadn't been nodding, it was over. Like, the decision was over, because even if every member of that city council disagreed with her, they were now backed into a corner where if they disagreed with her officially, they'd be massacred in the press by their constituents, because what story would have played better in the newspaper and on TV, right? And so my lesson, this is about 15 years ago, is that maybe it's okay to depart from my facts and figures, and maybe it's okay to try to set up an emotional connection. Now, I will freely admit that some of that emotional connection becomes manipulation. So the photos I can use, the words I use or don't use, and I don't make any I don't apologize for it, because my goal as a scientist is to affect conservation change. And so I found a book called Don't Be Such a Scientist. It doesn't say don't be a scientist. It says don't be such a scientist. And the goal there is you can manage your message and how you interact with people to be more effective. So if you haven't read it, it's a good book. I highly recommend it. So one of the first things that we ran into is what are we referring to? So the word feral cat comes out of my mouth very, very easily. That's what I've called it for a long time. But what we found is the word feral gets some disagreement, because some people say, oh, these aren't feral cats, these are my cats. Okay, do you own them and house them? Well, no. Do you feed them? Well, sometimes. So the word feral has this connotation of like in the wilds of the wild, right? What does a semi-feral, what does that mean? There's 100 people in here. I bet we'd get 20 different definitions of semi-feral, right? So the word unowned versus owned. In Texas, we have some interesting laws where you can do certain things with animals that are owned. And if they are personal property, then if I trap them or remove them, now I can be held liable because they're someone's property. And so we try to stay away from owned versus unowned, because just because someone claims they own them doesn't mean they're keeping them inside, doesn't mean they don't have an impact on the birds and the lizards and the small mammals. And then also I found that outdoor cat brings up connotation of a cat that lives inside 90% of the time and just goes in the backyard for a few minutes a day. But that's not what we're talking about. We're talking about cats that are outdoors a lot. So I don't use the word outdoor. And free ranging, when people hear free ranging, I ask them, what does this mean to you? And they say, oh, free ranging means in the wilds of the wild. It's ranging in the wild. And so I try to stick with the word free roaming, because it doesn't seem to conjure up connotations of pets. It doesn't get into the ownership or the fed status. It just is, if it has access to the outdoors, it is free roaming. And that tends to rile people up less. Now, you've seen this. This comes from Peter Morrow's book, Cat Wars. In the US each year, cats kill 2 billion birds, that's billion with a B, 12 billion small mammals, almost 500 million reptiles. That's probably wrong, not the same number of amphibians. Responsible for 14% of modern extinction. So in this room, this resonates, right? This resonates a problem. Other people don't care about this. So I have stopped putting up these words. It's something that I care about, something that we care about, but it doesn't resonate with other people. This does. Free roaming cats have shorter lifespans and lead really difficult lives. It is really sad for this individual. It is really sad for these groups of cats to be stricken with disease or hit by cars or to be poisoned by toxins in the environment. This is the message that I have found works better with people that are not coming from a bird, wildlife, or conservation background. Free roaming cats are vulnerable to many diseases and infections. So I don't apologize for using some of these photos. I use them carefully and judiciously, but I use them. Oh, there's also a few more. And so then we found that it works well to talk about the impacts to pets, right? Here are the pets that cats can transmit from wild animals to your pets. And we know that pets aren't receiving this many vaccinations. And so it personalizes it. Now, I have to cringe at my own self for personalizing, some anthropomorphizing, but this is the only message that I found that works. So the idea behind this local effort of Cats, Birds, and Us is, can we convince birders to partner with cat advocates to solve the cat problem? No. But can we get them, it's not showing it, but can we get them to agree to prevent permitted colonies in or adjacent to parks and natural areas? And maybe, yes, we can. Maybe we can find something that is palatable or less difficult for people. So we decided in San Antonio to start with fewer cat abandonments and say, as a birder, I want fewer cats to be dumped. As a cat advocate, you want fewer cats to be dumped because you're the ones that are feeding and providing vet care for these abandoned animals. So in this group, we wanted to create a map of sensitive areas in San Antonio. We wanted to calculate a standard buffer distance that we could go to the city and say, don't allow cat colonies to be established within X number of feet of these natural areas. We wanted to map out existing colonies, and we wanted to create a photo library so that we can make that emotional connection. Here are the cats. Here are the shape they're in. Here is the poor 17 cats at this location. Look at the filth of this location. Look at the amount of trash and raccoons. We wanted to get visual evidence of this stuff. And then we were going to use that to form relationships with and build trust with cat advocates, establish those mutual goals with them, petition the city to deny a managed cat colony permit near those sensitive areas. And then eventually the 50-year goal is to get San Antonio residents to treat cats like dogs. Don't abandon them, don't feed them, keep them on a leash. What we're not doing is declaring war on cats, saying how bad they are for birds. We're saying we care about the cats and we want to treat them humanely. So a little bit of math. I don't expect you to read this. I just want you to know that I have this and I will share it with anyone that wants it. Essentially, we took a couple of lit reviews that were already done. We mapped out what is that buffer if the home range was circular, and I know there's all kinds of problems with that, but we came up with a minimum buffer home range. If we were to buffer these natural areas, minimum of 82 feet in the literature, maximum home range would have made a buffer of 4,500 feet. And then we found the median was about 1,000 feet. And then with the 95% confidence interval, it was between 600 and 1,300 feet. So now we can say, okay, we have known endangered species habitat. whether it's a cave or golden-cheeked warblers, how far do we need to buffer the establishment of those cat colonies to prevent the impact to this particular resource? So we said, if we're feeling successful, it's 1,300 feet. If we're trying to scrape by with anything, we'll take 600 feet, okay? Then we got a GIS student volunteered to do some mapping for us. And here's a map of all known teeny habitat, threatened and endangered habitat in San Antonio. It also includes some golden-cheeked warbler habitat, some caves, endangered cave invertebrates, springs, streams, and then all of the San Antonio parklands. And then it failed. That's as far as we got. So the question is why? Well, we decided this was a very controversial issue, so we were going to move carefully and purposefully. And that's why. People got bored. We brought in Peter Mara, he fired everyone up. We have to do something about it. We had like 20 people in a room saying, I'm going to volunteer for this effort and solve the problem. So we set out goals to collect data and research and do some science, and it wasn't fast enough. And I was proud. We got all that done in 3 months, like GIS and lit review and all this stuff. So it took 3 months and they got bored. They said, well, I signed up to do something. Well, some of us were very busy, but not all of us. The other thing is the leaders were not expecting a long haul effort, which is funny because they said this is going to take a long time. So I thought they were in it for like 10 years. And after 3 months, they said, you know what, I'm going to move on. Okay, so we just had the wrong leaders with some unhealthy expectations. And then we also didn't have enough early wins. Had we built in, like with all those 20 people, these people do this, these people do this, and then we bring them together and show those early wins, maybe we could have kept them engaged, but just the volunteer management, we chose the wrong strategy. So what would I do differently? I would have done all that scientific research before assembling a large team. I would have waited to bring in the dynamic speaker until we were ready to pull the trigger. We had all the plans, all the maps, all the assignments ready, bring in a speaker and say, go do good things. And then we would have made sure that all those volunteers were constantly engaged. Now, many of you are saying, well, yes, Richard, this is clearly volunteer management 101. We just chose a different strategy. And it was the wrong strategy. So looking back on it and now presenting it with a title that said we failed, is obvious why it failed and what happened, but we just didn't predict it. I wanted to share just some main resources that we use. The Wildlife Society put out a package in the Wildlife Professional that was really helpful. Some good information, some good citations, and some good graphics. I mentioned Peter Mara and Chris Santella's book, Cat Wars, and then Stan Garrett, Seth Riley, and their co-author, Brian Cipher, the book Urban Carnivores, fantastic. Great lit review, great resources for home range, use that extensively. And then the last two is just a recommendation to see if any of your partners have position papers. The reason we were contacted is we actually have a position on trap, neuter, release at the state level. And so our partner said, look, we can lean on that to give us some political cover, right? This is not our idea. This is just what the state says it's appropriate. And so if you guys have partners that either have these or can develop them to give you and your other partners more cover, that would be recommended. And it kind of helps to broaden the circle of knowing that David Drake isn't off by himself with a personal vendetta against cats, because then David Drake's house will be, the address will be published. So, you bring in more partners. That's what I have. And if I have time, I'll turn it back over to Seth for questions.
So are you gonna do the new strategy?
If the right person comes along to lead it. One of the troubles, Seth asked if we're going to do the new strategy, and one of the troubles is that everyone knows exactly what Parks and Wildlife thinks about this issue. And so if we were to lead it, it would be a predetermined outcome. And we've learned that when we build the relationships, they have to be perceived as having a neutral or a open opinion. And if I were to call up the cat advocates, they'd say, well, I know exactly what you want to happen. You want to kill all the cats. I don't, but that's the perception. And so if the right partner comes along, absolutely.
I don't know, disseminating information with like neighbors, just like talking to each other, which obviously is as like coordinated and there's like action to it, but.
So the question was whether any efforts for like between and amongst neighbors rather than a coordinated effort. How do you grow that? I mean, that's a very grassroots thing. And if that ever happened, we would jump on it and support them. But if it's not that, then it's more of a recruiting, coordinated, orchestrated, less organic thing. And I'm sure that's happening on a local basis, but from a citywide or statewide. Yeah. One more, yes.
I'm curious between like these cat colonies and these maybe what we would consider unowned cats versus people who have personal pets that they let outdoors, who maybe view their cats having a right to be outside or a quality of life thing versus people who are looking out for these colonies. Is there a difference in like how you interact with them or what their feelings on these efforts are in terms of like, yeah, based on those different types of cats?
Yes. So the question was, is there a difference between people who are just letting their cat outside on a limited basis versus people who are managing cat colonies? It's a message of personal responsibility. The people who say, oh, I just let my cat out for a little bit, they're saying that because they have that personal responsibility and they know that it's potentially a problematic thing. The other folks that are saying, this is what I'm doing, this is right, and this is the entire meaning of my entire life, it's a different conversation. And so with the personal responsibility, my response is, well, let's talk about how you treat your dogs, right? And can you bring in your cat more often? Or can you build a catio, right? With the folks that are managing cat colonies, I talk about diseases and I talk about the photos I get from the police where cats are chopped in half. And I'm having as a biologist to investigate this from animal cruelty, but I know that it's actually coyotes and owls that are eating these cats. And so I show them photos with their permission. And I show them photos of the diseases because that's where we're going to get the diseases and the mange and all that stuff. So it is a very different set of vocabulary. Yeah.
All right, let's say we can all of our speakers. Hopefully with some of you, see some of you back at 3:30. I think Urban Habitats is in here.
I just pulled the wrong thing out. That was a one-time move.
Oh, okay.
Yeah. Where's the Go up and stay and turn right. It's in the front. Okay.
Because I really want to, I mean, we talked about patio.
Yes.
And
Yeah.
those things. It's just not, I'm just afraid. I'm so afraid when it's bad. I think one of my people keep coming up to me being like, it's so much. But I just have to read up this for the next session if you want to chat. Everything you're saying. Are you good with PowerPoint? Yeah. So the fun part about this, fun part about what we're doing here. No, we're trying to